still invested.
I'm sure you have.
But the network's out there. So
I'm at a base.
Dodger, California, New York, both watch. Well, like you're talking about people that's going to cost probably
Hey, good afternoon, everybody. Welcome. We'll just get started here. So we have some exciting talks coming up, plus also the election day. Nothing will be held that day. Just a quick reminder, there'll be other exciting things going on. Keep us busy. And without further ado, I will introduce Dr. Young, to then we'll bring up our speaker, Dr. Kelly. Thank you all.
All right. Well, I'm going to keep my comments short because we all want to hear from Dan. But rather than go through his bibliography and CV, I guess I'll just distill it down to 2 sentences. One is that Dan is probably the best thinker, scientist, most creative scientist in the field of heart metabolism that I've ever encountered. And what distinguishes him is the creativity, the rigor of his science, the meticulous attention to detail, and then the even-handed approach that he really is trying to find the truth in a way that is uncompromised. And so he's been an inspiration for me, and I think for most people in the field of heart metabolism. So with that, Dan, welcome. We're delighted to have you.
Well, thank you. Can you hear me okay? Thank you, Larry. And it's really been great to be here. I've spent 2 days here, and it has a great feel, high quality science and collaborative environment. And every time I come back, I'm really more impressed. So you should all feel really good about that. I'm going to tell you a story today, and then a short story at the end that's linked to it, but one that actually started when I was a cardiology fellow to a certain extent, as I'll touch on very briefly. And I thought that would be good in part for the trainees. These are my disclosures. They really don't relate directly to what I'm going to talk about today. I really don't have to show this slide to this group. It basically indicates that heart failure is a horrible disease. It's actually, quote, malignant. If you look at their survival curves, it ranks with many cancers, malignant cancers, of course. And what's interesting, or maybe disturbing, and it was true when I also was doing my first research project as a cardiology fellow, is that in contrast to the cancers, the treatment for heart failure is largely just kind of a one-size-fits-all. Now, that's a little unfair because we have now defined these 2 phenotypes, reduced ejection fraction and preserved, but it's really still true. And more importantly, we're not treating at the root primary drivers. And so I became interested in looking at and letting genetics identify pathways that might lead to heart failure and cardiomyopathy in relatively rare inherited diseases to see if we could get any clues and move forward. And as you may know, one group among others is inborn errors in mitochondrial energy production, many different steps that cause cardiomyopathy. Of course, there's also genetic defects in the sarcomeric apparatus, and we were looking at that also at the time, and others. But obviously, this looked to be an interesting pathway. And way back when I did this, we focused on inborn errors in mitochondrial fatty acid oxidation that afflict children who develop heart failure. And the details on this slide are really not so important, but LCAD, MCAD, LCHAD deficiencies, these are deficiencies in beta oxidation enzymes that cause metabolic disturbances, including hypoketotic hypoglycemia, but under stressful conditions like viral illnesses, when they would fast, they developed heart failure. And it was a striking thing, and it was a great project for me because I also learned how to clone genes. Back when we actually had to clone genes, it was before PCR. I'm sorry to say, during the context of the project, PCR came on age, so that helped accelerate things. So it was a really good project for the toolbox, but it was also important because it led me and others to start thinking about whether we're learning anything from these fatty acid oxidation defects in acquired, more common forms of heart failure. And this leads me to a slide that's kind of a primer, if you will, for the cardiologist on the fuel metabolism in the heart, because obviously this led to an interest, long-standing interest in fuel metabolism. And as shown up here, there's a really interesting transition between the fetal stages to the adult that I talked about actually yesterday to the Vascular Biology Institute group. And what happens is, in addition to an expansion of mitochondria, is that these mitochondria specialize and become poised to oxidize fatty acids at very high rates, which is the chief source of ATP in this very high energy requiring organ. Okay, it's not to say that glucose isn't important, lactate and other substrates, as you'll see, but fatty acid oxidation is very important, and this is underscored in a way by the genetic defects that these children suffered from. But importantly, perhaps more importantly in some ways for this talk, is that during the development of heart failure and pathologic cardiac hypertrophy from a myriad of causes, ischemic insult, hypertension, genetic forms of cardiomyopathy, this fatty acid oxidation machine becomes deactivated. And so this high capacity for fat oxidation goes down, and early on we found that this happens at least in part at the transcriptional level, although there's post-transcriptional levels. And so the expression, the high expression of these enzymes in the mitochondrion, the very enzymes that the children had genetic defects in are downregulated. And an early part of heart failure, and this at the very least was correlative, but there was also evidence that in parallel, phosphocreatine stores were going down and predictive of outcome. So we got really interested in this, and it led to really a career-long interest in trying to understand how this happens and whether we could intervene in some way to begin to add to our therapeutic tool chest metabolic modulators, maybe that would be added along with heart failure. And again, getting back to, as you all know, heart failure, it's not that it's primarily driven solely by these changes, but we know that there is kind of a cascade of pathogenic drivers. Sometimes inflammation is greater, sometimes perhaps these metabolic, but ultimately it's a common phenotype of heart failure. And we wanted to be able to address multiple of these and add to the tool chest. So what I'm going to talk about today are two things, and these are, in a way, strategies to rebalance fuel utilization in the failing heart. Mostly what I'll talk about today is the work that we've done that identified an alternate fuel that could be useful in heart failure. And we started at very fundamental, and I'll take it as far as I can into the more translational studies. And hopefully there's time at the end. We have a short piece of work on that I think you'd be interested in. It was recently published, actually, on looking at SGLT2 inhibitors. It's just, as Larry and I were talking about earlier today, a piece of the puzzle. Not saying it's the answer, but there's so much interest in this. And the question of whether they also could work as metabolic modulators comes up. So let's start with the ketones. So this started maybe about 6 years ago, 7 years ago, maybe now. We were down in Florida at the Sanford Burnham Institute. There was a group, Greg, Rick, and Julie. Greg, an MD, a PhD, now cardiologist, a faculty member and on to industry, but at the time came over from Switzerland and was interested in doing metabolomic and proteomic profiling of early stages of heart failure to learn more about what was really going on. And Rick and Julie were also involved in this in various ways. The second part of the team was really did this at Penn more recently, Micha, Tim, and Fawaz. This is published, so I'm going to summarize it. What we did was we took a mouse model of heart failure, which is pressure overload combined with a small apical myocardial infarction that results in a consistent LV global dilatation, isolated mitochondria, and subjected the mitochondria to targeted metabolomics and quantitative proteomics. And we found some interesting things. Again, I'll summarize. We found that two enzymes involved actually in ketone oxidation, BDH1 and SCOT, were upregulated in the early stages of heart failure in the mouse model. And in addition, as is shown down here, there were a number of short-chain acylcarnitines that were readouts for CoA moieties that could be produced by high rates of ketone oxidation. It could also be produced by other factors. When we put these things together, it strongly suggested that there was ketone utilization. Now, unbeknownst to us, at the time, there was a separate group up at the University of Pennsylvania, led by Ken Margulies, Ken Beatty, Nate Snyder, and they were doing very similar profiling of end-stage heart failure from humans. And as you may know, Ken has this massive biobank that he's collected from heart failure transplant recipients, and then donors that have normal function, but for one reason or another are not suitable for donation. And amazingly, one of the few times that I've seen such strict correlation between mice and humans is that they found essentially the same thing, although I should mention that their metabolomics was CoA-based, not carnitine-based. That's what Nate Snyder does for a living. And it was it was more unbiased. But they really found the same thing. Ken called me, and we kind of excitedly compared things over the phone. So this led to the possibility that in the context of reduced capacity and utilization of fatty acids in the failing and hypertrophied heart, that ketone bodies, 3-hydroxybutyrate and acetoacetate that are made in the liver were actually providing potentially an ancillary fuel. How much of this was going on and whether it was adaptive, maladaptive, or of no consequence whatsoever was not clear at that time. We have a much better idea now, and I'm going to summarize a number of published studies that largely came from our group. First of all, in studies in which we used 13C-based isotope tracers in isolated hearts, we found that failing mouse hearts increase utilization of ketone bodies, okay? In addition, we found out that disabling cardiac ketone oxidation in the mouse heart by knocking out the first enzyme in the heart that oxidizes ketone bodies worsened heart failure in these models. It actually also gave a really interesting fasting-induced cardiac dysfunction. Happy to talk about that as well. Thirdly, as I'll show you in a moment, infusion of beta-hydroxybutyrate, the main ketone body, the most stable ketone body, reversed cardiac dysfunction and hypertrophic and pathologic remodeling in a canine tachypacing model of heart failure. And in chronic studies, in collaboration with Dan Westenbrink's group in Europe, we used oral ketone esters as both preventive and treatment strategies in mouse and rat models. So taken together, number one, this suggested that this switch, if you will, to ketone bodies was indeed adaptive. And it raised our interest in a couple of things. One, could this lead to any kind of translation? And secondly, how the heck do they work? So just some data of what I just told you. This is a bizarre model in a way, but it's also quite amazing in that it follows in 1 month's time the stages that human heart failure goes through and over years in most cases. And this is a tachypacing model in the canine model. And by 4 weeks, as you can see, basically in the green lines here, up here, is that there is a progressive drop in cardiac output, ejection fraction, rather massive increase in left ventricular end diastolic pressure, number of other things. And we began a ketone infusion midway through, right when things start going downhill. And as you can see, there's a rapid amelioration, in some cases even improvement over baseline of what we saw. So this was the most dramatic response I had seen in heart failure models and really piqued our interest. We also, as I've mentioned, have gone into mice and rats using various MI models or TAC models. And we have fed these animals ketone esters. And now you can't give animals or humans for that matter ketones. They taste horrible. They're sodium salts, they're acidic. But what we could do is provide this C6 ester, okay? And we have both mono and diesters, which is basically to take the ketone body, 3-hydroxybutyrate, and put on two, in this case, 1 or 2 medium chain fatty acids. And then in the liver, and probably also to a certain extent in the gut, these are cleaved off. And what you're left with is the beta-hydroxybutyrate. This cleaving probably occurs mainly in the liver. And then these medium chain fatty acids also drive ketone production. And so this raises levels of the ketones upwards 1 millimolar or even more, 2 to 3 times levels of what is normal. I should mention, interestingly enough, that the half-life is so short that the elevation of ketones in the animals was mainly only during the feeding phase. Okay, so this is at night, of course, for the mice. but then they're fasting more. So, but we probably were only getting coverage, exposure, if you will, for half the day. But nonetheless, as you can see here, and as is published, we had a pretty nice response in terms of reduced remodeling and increased function when we gave these ketone esters. So we're doing these studies, and we began to wonder whether any of this was relevant to humans. And I'm going to talk a little bit about not just work that we've done, but work that others have done as well to demonstrate the importance of ketones at time to the heart. The first thing that I'd like to bring up is probably a paper that a number of you have seen. And if you haven't, it's really a fascinating paper by Zoltan Rani's group, who's down the hall from me, actually. And there's a lot of data in this paper, okay? But what he did was to work with the cardiac electrophysiologists at Penn, who were doing AFib ablations. And of course, they have catheters in the coronary sinus, in the aorta, peripherally, and they can then take samples, okay, and we can do targeted metabolomics and look at the extraction fraction and relative contribution based on that to ATP production. It is a bit of a prediction. It's not measuring oxidation per se, okay, which is important, as Jerry certainly knows, but it's still quite a tour de force in humans. And I'll refer to you to some of the color coding here. And what they did was to look at those individuals getting the AFib ablation with normal function, and then, of course, those who had heart failure. And those are compared here, preserved ejection fraction, which is not heart failure, probably was not the right terminology to use, but it's normal cardiac function. And I refer you to the yellow and the red slices of the pie. Number one, and as I've told you, there's a diminution in the oxidation, or a contribution, I should say, of free fatty acids to ATP. Now, this is relatively modest, okay? It's in a fasting state. And it also kind of emphasizes the point that the heart doesn't stop using fatty acids. Its capacity goes down. And certainly if you stress the heart, it cannot increase the utilization of fatty acids, but they go down. And importantly, what went up was ketone bodies. And that's shown in the red slice of the pie. And the other thing that's interesting is if you look at the graph on the far right here, that this is showing the extraction. It's in inverse, but basically the concentration of the beta hydroxybutyrate dictated the amount of beta hydroxybutyrate that was being taken up in the heart. So the heart will basically eat as much ketone as it can get. And as I'm going to show you now, it turns out, interestingly, that in human heart failure, levels of beta hydroxybutyrate, circulating levels, increase in the context of heart failure, okay? And so this has been reported. We weren't the first to report this, but this is done actually by a medical student who worked basically evenings and weekends, didn't have time off to do this, Fawaz Naeem. And this was also done in collaboration with Dan Rader's group, in which he has enrolled, as you may know, a significant number of individuals over the years that have excellent genotyping, clinical phenotyping data, as well as plasma for us to analyze. And we did this in collaboration with Pfizer because they were interested more in we could talk about this on the side, but more in the branched chain amino acids and that were present because they were interested in this linkage, a different topic, branched chain amino acids and heart failure. Happy to talk about that. And the bottom line is shown here, interestingly, that in the heart failure and I should have mentioned, we had over 200 normals, heart failure with preserved ejection fraction, heart failure with reduced ejection fraction. And as you can see in the green bars, the levels of 3-hydroxybutyrate and one of its metabolites, butyryl carnitine, is shown in this pathway, were elevated in the patients with heart failure with reduced ejection fraction. They trended towards being elevated in the heart failure with preserved ejection fraction, but this was not significant. This is, to my knowledge, the first comparison that was powered between these. It needs to be replicated. There have been other studies, as I've mentioned, with heart failure with reduced ejection fraction, small numbers of patients that have high circulating levels of ketone bodies. The other interesting thing about this is that this is kind of a real world analysis. In other words, there's not a controlled fasting or anything here. So it's amazing with all the noise that one gets, just, patients that had either just eaten or had been fasting for 16 hours, didn't matter, we were still getting this signature. We had a number of other interesting signatures that I'm happy to talk about. Again, a topic for another time. But the point here is that indeed there is a heart-liver communication in the context of heart failure. We also did targeted O-link proteomics on this, and looking for proteins that might be correlated with this to get an idea of what might be signaling some of this. And we still don't know, but I'll show you the correlative data. And basically what we found was that NT-proBNP, which was higher in the HFrEF versus the HFpEF patients, which has been shown by many groups, correlated with levels of, as you can see here, the ketone bodies. So this was interesting. I should also mention, and these kind of complicated plots, but for those of you interested in fatty acid oxidation, we and others also found in the heart failure with reduced ejection fraction group that there were higher rates of circulating long and medium chain acylcarnitines, suggestive that some organ it has a high flux through the fatty acid oxidation pathway. Again, indirect evidence, not direct. And I should mention that it's not the heart. The heart has lower flux through this. So this also suggested, along with the previous data, that the liver was receiving these signals to crank out, if you will, ketone bodies and probably fatty acid oxidation, which often go in parallel when there are free fatty acids that are increased in bombarding the liver. And so one model we have, which is a testable model, but is not proven by any means, but we're intrigued that there are some mediators that are secreted from the heart, hormonal or otherwise, one of which could be BNP and ANP, which are known actually to increase lipolysis in adipose tissue, as shown by a number of groups, including Sheila Collins and others, and that the high levels of free fatty acids, which have been shown in heart failure, are driving the ketogenesis and the fatty acid oxidation. But this, of course, needs to be further tested. This has led a number of groups, mainly in Europe, to launch trials, human trials. And so I'm just going to kind of briefly tell you about those. So the first trial that came out was patients that had severe acute heart failure upon chronic heart failure. And what they did, this is Henry Wigger's group. The group infused beta-hydroxybutyrate, and basically what you can see is the hemodynamic changes that occurred rapidly after infusion. And this includes an increase in cardiac output, a decrease in wedge pressure and systemic vascular resistance, an increase in ejection fraction. The results are quite startling, and they're quite rapid. And this has led many of us to propose, or at least it's a quandary about how this is working so rapidly. And I think some of the cardiologists in the room will wonder whether this is really a drop in systemic vascular resistance, an impedance mismatch, versus a primary effect on the heart. And it turns out that ketone bodies have been shown to cause a drop in vascular resistance. So whether this is a primary drop in vascular resistance with a secondary increase in cardiac output versus the other way around, direct effect on the heart, we just don't know yet. We have evidence for both, to be honest. What we can conclude is that this is a pretty effective approach for severe heart failure and acute treatment. The same group recently, in an unbelievable study, I don't know how they got this through IRB, but they brought patients in, maybe because these patients were on death's doorstep, but they brought patients in cardiogenic shock, and they infused ketone. And as you can see, the ketone, the circulating ketone levels went up, concordant with the increase in cardiac output, the same kind of results that I just showed you. But these are really sick patients. And again, for the clinicians in the room, you know that oftentimes the problem we have with this is that if it's a vasodilator, it's a disaster because we can't keep the blood pressure up. If it's an inotropic agent, it looks a little bit like this, and then we have trouble in the long term. But somehow, amazingly, the blood pressure is being maintained. So this speaks a little bit more for perhaps an inotropic action, but we just don't know. The only study we have now for ketone esters, chronic heart failure, are with these ketone esters. They're given in like a nasty tasting shake, like a milkshake. And this is another amazing, this is the same group again, that actually was able to start them on a 2-week regimen. It's a randomized controlled double-blind trial. And also somehow did hemodynamics on these patients. Okay, so it's again an impressive study, I guess. It's really quite modest, to be honest. If you look carefully at the y-axis here, you'll see that although there is some response, in this 2-week period, it's pretty meager, to be honest. And so either this treatment wasn't long enough, you know, for the chronic effects, or it's just not going to be useful for the most part for chronic heart failure. This is kind of where we're at right now, and they're doing longer term studies to see if it makes a difference. And should also mention that I often get this question. It will be a tough treatment. It seems as though it would make more sense if we thought it was going to be a powerful therapy to figure out a way to drive ketogenesis from the liver. There's a ways to go. Obviously, already the ketone levels are high, but we have room to go there. But that's also going to be a tricky business. But with all of that said, the acute heart failure studies have been impressive. The chronic ones remain to be determined. So how do they work? You know, the way I've presented this kind of makes it sound like a done deal, but by no means. And in fact, the reviewers of most of our work, the very astute reviewers in metabolism and cardiac physiology, brought up the possibility that this is not just a fuel supply. You know, after all, fatty acid oxidation isn't going down all that far. But on the other hand, we have good evidence to suggest it is being utilized. As it turns out, there are other mechanisms. And this is not simply only work by us, but there are many groups, Eric Verdin's group, a number of others that have shown that in addition to providing a fuel to the failing, starving heart, that there are myocyte and other cell type fibroblast protection, anti-inflammatory effects, signaling effects, epigenetic effects that could easily be at play. And as I've brought up, the vascular resistance aspect could also be at play. So it raises a very fundamental first question. Can we demonstrate that ketone bodies have a direct effect on a failing myocyte? It sounds like a simple question. It's not so. But again, in collaboration with Ken Margulies' group, who is isolating myocytes from hearts that are taken out from transplant recipients as well as donors that can't be used, we could look at the effects of ketone bodies on contractility. And that's what I'm showing you here in a rather Herculean study that typically occurs in the middle of the night. But what this shows is a number, and it was published, so I refer you to the Jack BTS paper, but let me kind of walk you through this. This was isolation of myocytes, putting into the ion optics, looking at contractile function, so you can look at both rate and degree of shortening, okay? And you can see up here that the normal, on the bottom, the normal myocytes from donors have a greater degree of shortening and relaxation compared to the failing myocytes. And then what we did was we cultured them and we only put them in a kind of a typical amount of glucose without any other substrate. And then we added substrates in, first fatty acids, first beta hydroxybutyrate, free fatty acids in dose ranges and together. And it had no impact on the normal myocytes. They contracted fine either way. However, when we did this with the failing myocytes, as you can see, either beta hydroxybutyrate or free fatty acids had a significant impact on the degree of shortening and relaxation. So this was interesting to us. It indicated that indeed the ketone bodies, as well as fatty acids, because there were no fatty acids there to start with, are helping contractility. It also indicates, if you think about it, that probably the utilization of glucose alone, perhaps glucose oxidation, which has been proposed in heart failure, is altered so that the normal myocytes can, if you will, contract reasonably well on glucose alone, but glucose can't do it. Its utilization is also altered. So anyway, for those who think a lot about this, this was evidence indeed that the ketone bodies were acting directly. So we also have some evidence that ketones work independent of ketone oxidation, at least on the remodeling. And what this shows is that when we were doing these ketone ester studies, not only did we improve function in the rodent models of heart failure, but we also reduced the pathologic hypertrophy. And we were interested in studying this in culture to see how that worked. And so what we did was to isolate myocytes from, in this case, normal mice, adult mice, and then we subjected them to a hypertrophic agonist, phenylephrine, with or without ketone body treatment. And when we did that, as shown up here, here's the hypertrophy is measured by width to length. And then here is the ketone bodies, either with or without phenylephrine. And you can see that the phenylephrine effect is abolished by the addition of the ketone body. Then we repeated this experiment in mice that had a knockout for BDH1 that can't oxidize ketone bodies. And we still had this same remodeling effect, suggesting that, and this is early stage data, but suggesting that this effect occurred independent of ketone oxidation. We also did this with both the R and S stereoisomer, because the S stereoisomer cannot be oxidized by the pathway, and we got the same results. So suffice it to say that based on these two sets of experiments, we need more to do it, all unpublished data, that there is evidence here, I believe, for the ketones working independent of ketone oxidation. So I would argue, although I guess we haven't directly measured the vascular resistance, that there's likely, it's not completely satisfying, but they seem to have pleiotropic effects. And there's a whole body of literature emerging now indicating effects on fibroblasts, on endothelial cells. Stay tuned. A lot of this work is coming out. So it's an interesting area that needs essentially more work. So for the last part of this talk, I want to tell you about what was kind of, in a way, a humbling experience, but we boldly wanted to ask how these incredible new therapeutics for heart failure, which weren't made for heart failure, the SGLT2 inhibitors, how they work. I don't know how they work. I can tell you that much right now. I can show you a piece of the puzzle that was surprising to us. There are some caveats, and I thought maybe it would stimulate some discussion. So first of all, you're all aware of this. But these SGLT2 inhibitors were developed to lower glucose in diabetics by inhibiting the sodium glucose transporter in the kidney. I remember being on several panels when pharma would ask us, I wasn't alone. I think one of them was myself and Helen Hobbs and Mitch Lazar. And we all thought this was the most ridiculous thing in the world to do something like this and have people peeing out glucose all over the place. And as you probably all know now, these are very effective glucose lowering agents. With all glucose and anti-diabetic type of agents, they need to go through cardiovascular outcome trials. And again, as you all know, the rest is kind of history. Not only were these safe, which is all that they were trying to find out, is that the rate of hospitalization and to a lesser degree, but significant mortality from heart failure was reduced more than any standard heart failure drug that we had on the market. And so this is incredible. It's been replicated many times. It's also been shown, but I would say somewhat less effective for heart failure with preserved ejection fraction. And one of the things that piqued our interest in this is, and this is something on a little supplemental thing of one of the other EMPA-REG related studies, but it's known that SGLT2 inhibitors increase circulating ketone bodies. So we got all excited about that. And the way that they probably do that is in large part changing the insulin and glucagon ratio and really bringing insulin levels down, driving some degree of lipolysis, at least that's the model, and which in turn drives the liver to produce ketone bodies. So Justin Berger, a pediatric cardiology fellow in the lab, in collaboration with Sap Halder in the Amgen group, we wanted to ask the question, well, we really were starting this at any rate to try to figure out what organs were responsible for the cardioprotective effect downstream of the SGLT2 inhibitor. Justin, by the way, has now taken a faculty position at WashU. But he kind of commandeered this study. And the approach was we had a number of different knockout lines. One was generalized SGLT2 knockouts. The other was cardiac specific, which was kind of silly in a way because there's hardly any SGLT2 there in the first place, but we needed to do this. And then an inducible kidney knockout. But we started with the generalized knockout, and as you'll see, we didn't continue for the reasons of what we found. The first thing we wanted to do, and I'm showing the wild type animals on the top that are being treated with EMPA, and then the knockout at baseline. Okay, we wanted to really do pretty robust metabolic phenotyping of these mice, because one of our hypotheses at the time was that SGLT2 inhibitors are reprogramming substrate utilization. And I think that's still a viable hypothesis, although you'll see why the waters have gotten a bit muddy. But as you can also see up here, in both cases, really the generalized knockout phenocopied the effects of SGLT2 inhibitor given to the wild type. So look at top, there's a drop in glucose. There is indeed a ketogenesis, massive spilling of glucose in the urine. Body weight, it changed a little bit more with the knockout. It was a modest diminution. So these are effects, of course, that we see with inhibition of SGLT2. So we felt good about that, along with the fact that we had actually knocked it out, which is a good thing. We did some other, types of studies. Targeted metabolomics, it's what we do. Again, if you look at the top, a variety of myocardial acylcarnitines are actually increased both with the EMPA therapy and with the generalized knockout. There's also an increase in a subset of TCA cycle intermediates. We're not sure what to make of that, but it looked like there's something happening, certainly. It's interesting. The pattern of increase in the TCA cycle intermediates is a little bit more reminiscent of an anaplerotic effect rather than a full cataplerotic effect around the TCA cycle. So then we spent about, this is probably important, we spent about a year trying to optimize EMPA treatment of mice and heart failure. And mind you, there's very little data in humans that SGLT2 inhibitors actually change remodeling. There's some MRI data, and it moves in the right direction. But the best data we have are hospitalization rates, exercise activity, and mortality. So we became worried that it doesn't work at all in the remodeling in the first place, and we couldn't use that as a readout. But we did a number of different heart failure models in mice, and at first we had trouble showing effect in the normal mice. Then we had to go back and redose, and along with Amgen, we found a dose at 50 milligrams per kilogram per day in chow that actually gave a reproducible benefit in basically the mode that's shown here, which is given the SGLT2 inhibitor, as shown in the top left, 3 days after the TAC-MI. And this also is interesting because you could argue this is not a treatment mode per se, but maybe a preventive mode. It's kind of in between, I guess. But again, the studies in the humans are also, they're already on SGLT2 inhibitors, so we thought we would at least mimic that. And I'm not showing you all the data here. You have to take my word for it that it was working in the normal mice. It's in, we have that data in the paper, but it's so complex when I show it here. But I want to show you the key points here. The key points, first of all, are that with this TAC-MI model, if you look in purple, we're getting a pretty sick heart, okay? And there's, I'm sorry, the purple is in sham, and in the knockout, we're getting a pretty sick heart. And if we compare this basically to the wild type, it looks exactly the same. So the knockout of SGLT2 does not afford protection against this heart failure model in mice. In contrast, we do see the protection when we give the EMPA on top of the knockout mouse. Oops, I'm sorry. So basically, we're getting the same effects that we saw in the wild type mice, modest but significant in the knockout. And so this suggested to us, and we had to repeat it many times because this is the type of study which is in many ways, I guess, negative, you would say, to the hypothesis that is always hard to convince ourselves as well as the reviewers. But we've done it enough times now to say that basically genetically knocking out SGLT2 versus pharmacologically treating SGLT2 give different results. And if we give the EMPA on top of the knockout, we still get the beneficial effect. So we have concluded the following, that SGLT2 inhibitor treatment and the knockout exhibit similar metabolic phenotypes, okay, including glucosuria, ketosis, metabolomic evidence of changes in the fatty acid oxidation, and TCA cycle. The SGLT2 knockout mice are not protected against heart failure, at least in the model that we used. The SGLT2 inhibitor afforded similar cardioprotection in wild type and SGLT2 knockout mice, strongly suggesting that at least in mice, the actions are off target. I think as Larry had mentioned at the beginning, you know, we talked a little bit about this in terms of, I don't know, being careful, I guess, about the results. And I've learned over the years that, you know, obviously the mouse models can be problematic with heart failure. So I insisted with Amgen and with the fellow that we put mouse in the title. I'm not sure what this definitely means for humans, but it's intriguing. It's also interesting that there have now been two published, well, one bioRxiv and two published studies indicating that this is off target. The first two on the left were in mice. The one in the top, this is by Kurt Zurbier's group, believes it's cross-reacting with the sodium hydrogen exchanger. This is a controversial area because he's shown that the compounds interact and others have not. The one under that is one that really brings in an anti-inflammatory effect, which is interesting, but again, not on target. The most provocative study is one down the hall from us via Zoltarani's group, okay? And he has been working hard on this. He was also part of the Amgen thing, but as you could see with the results, he had to get out of the Amgen thing in a hurry. So basically what they have found, largely using a metabolic readout, not a functional rescue readout, except for with isolated myocytes, is that they believe SGLT2 inhibitors activate pantothenate kinase, pan kinase, which is involved in CoA biosynthesis and salvage, which in a way would make sense, and they're pursuing this. And they're working on showing the efficacy work. So I think this is where we're at right now. I find it to be fascinating that we are really spending a lot of time now working on trying to understand something that was first identified in humans instead of the other way around. That's unusual for our group, but we at least know it's very important, and I think it's a very important part of the field. We're also doing studies with heart failure and preserved ejection fraction. We're doing that in collaboration with David Leffer's group, using the existing HFpEF models in mice. I actually think what we need for the field is to start moving into large animal models and start doing that as well, because I'm a little worried about whether we're recapitulating things. But at any rate, the data are the data, and I thought this group would want to see it. We did publish this rapidly as a letter in Circ Research, and we'll see what happens. So with that, I think I've mainly identified the primary scientists involved, and our collaborators are shown here: Ken, Debbie, Peter Crawford, really critical for a lot of the ketone utilization studies. Bill Poo actually was more involved in some of our genomic work, and Doug Lewandowski. And with that, thanks again for inviting me. It's been a great couple of days, and thanks for your attention.
Thanks, Dan, for a spectacular talk. Very interesting and thought-provoking. So questions from the audience? Fadi.
So I was curious about the cell autonomous effects on contraction from the ketones and free fatty acids that you had in the heart failure model myocytes. Do you think that's related to improved calcium cycling, or is it
I'm sorry, I missed the first part of the question.
the effects of ketones on the isolated myocytes.
Yeah.
And is that related to calcium cycling or to myofilament sensitization to calcium, and the fact that it's only evident in the heart failure myocytes and not in the normal myocytes, which is very encouraging.
I can tell he knows David Cass. Asked me that. So it's interesting you asked that question. So those of us interested in trying to affect the energy-starved myocyte might say that it's providing ATP for calcium uptake and cycling. David Cass has seen these data, who, in my opinion, is one of the world's experts in understanding, EC coupling and hemodynamics. And he doesn't buy that it can happen that quickly from an energetic effect. And to your point, believes there's something happening with EC coupling or calcium. And we don't know the answer to that. We have been interested in using some of his models in that regard, but we haven't done those studies yet. So it's a great question. The other thing that we haven't been able to do, which we want to do, is to use myocytes that have a knockout for beta hydroxybutyrate oxidation, BDH1. In that case, if it still worked, it would be more a direct thing. But this is a weird thing, but the only myocytes we can get cultured that actually have a defect from the failing heart are either from humans or from large animals. If we do it in mice, they work fine. So we don't have dysfunctional myocytes in the first place. So it's been frustrating.
Dan, thank you. That was really wonderful and provocative, that last part. So a question and actually two questions. So, you've convinced me clearly from your studies and others, giving ketones back promotes cardiac contractility and can rescue heart failure. Two, we know SGLT2 inhibitors do promote ketogenesis. So your third conclusion in the mice, maybe the fact that you missed it in the mouse is the ketones. Maybe, again, mice are incredibly, as you know, hypermetabolic. They can shift. So did you, the first question is, look to see if ketones then tracked maybe with the inability for the rescue with the SGLT2 inhibitors, and the second, or with the knockout. And the second thing is the other possibility. I think you've convinced me that oral ketone supplementation is just not going to be practical in the patient. We know those issues. But what about something like an ACC2 inhibitor to promote endogenous ketogenesis?
Yeah. So the last question. I've been talking with Rong Tian about this, who is working with others. Then these things exist, which would increase oxidation, fat oxidation in the liver, drive ketones. I guess we have to think about the safety range here, just like the discussion we were having earlier. But I really like that idea. I also kind of like the idea of that Naseritide had failed. This is a longer acting natriuretic peptide. But if we could go back and look and see if we're driving ketogenesis, although again, it's not an oral prep, but people are starting to work on that. You are right that it could be a mouse problem with the ketones. And it is true also that when we look at ketogenesis from SGLT2 inhibitors in mice, it's only during the fasting period. With that said, with the knockout of SGLT2, we still got the benefit. So it doesn't rule, maybe we haven't ruled out a ketone mechanism because it wasn't the right milieu to do that. But the second part result is what really suggests that maybe it's off target, at least in mice.
Over here. Lovely talk. I want to propose maybe another way of thinking about how the ketones might be altering the cardiac metabolism, and besides just providing acetyl-CoA and NADH. And that's the fact that to get it onto a CoA, you have to take a CoA off of succinyl-CoA. And that bypasses a crucial step in the TCA cycle that generates GTP. And why I say that, it fits with two of the data that you showed, the AV difference showing increased amino acid uptake and the evidence of increased anaplerosis, which through the Charlie-Stanley pathway should be activated.
So in the model you propose, which direction would the GTP be going if the CoA is being,
It'd go down.
but what do you think that would, what impact would that have on contractility then?
Well, that would feed metabolites into the TCA
Yeah,
cycle.
that's a good point.
And provide a larger pool to enable more efficient.
And how,
cycle.
why would it be feeding, just for the rest of the group in terms of, and myself as well, why would that feed in more anaplerotic?
So two ways. One is by lowering GTP, you lower any inhibition on GDH, which is the only way you're going to oxidize your amino acids. You have to get rid of the ammonia. That's the key step. And the other is it is also reducing a cataplerotic pathway through PepsiK, which consumes the GTP.
And do you think there's enough of both of those in the cardiomyocyte to matter, I guess? I know at baseline, there's very little glutamine input, but you're right. Maybe in the failing heart, this could be much different. So it's something to be considered. I like the thought.
I mean, the AV extraction was about double in the failing heart.
Yeah,
of
that's a
amino
good
acid.
thought.
So-
I should also mention that there's yet a third model that Deb Muehl has put forth, and that we've been working with her on this, and that is that it can actually work in reverse, and it can shortcut the bottleneck that occurs in fatty acid oxidation that sops up all the CoA, and those two aspects release the CoA and change the redox back normal. So this is not an energetic issue, it's an unclogging of the beta oxidation pathway, which maybe makes more sense because, as we all know, the reserve in mitochondria, even in the failing heart, is quite immense. So maybe we oversimplify this notion that it's truly an energy-starved heart so much as a heart that's all gunked up, which ultimately will starve the heart, but.
Oh my gosh, this is a much less sophisticated question, right? Two great talks, Dan. Thank you. Why do you think that serum ketones are not elevated in HFpEF whereas they are in HFrEF? Does that tell us anything about the trigger to ketogenesis or liver production of ketones? And I guess the extension of that question would be patients who have sarcomere mutations, who have hypertrophic cardiomyopathy, maybe without clinical heart failure, do they have elevated ketones? So I'm sort of trying to dissect the hemodynamic issues versus something about the nature of the heart failure.
Well, I mean, we haven't proven it, but if we go with the model of lipolysis that's related to, BNP and AMP release, in general, the AMP and BNP has been higher, at least in our surveys, in the HFrEF versus the HFpEF group. So if that follows, we should get a subset of HFpEFs that have, typically high BNP compared to HFrEF. We haven't done that. And I don't know, it's a good question about hypertrophic cardiomyopathy. I don't even know what the AMP levels, et cetera, but that's a really good question. And given that Charlene Day is down the hall, I should do something. It's a great question.
As I'm walking, what about catechol in heart failure? They're clearly
Yeah.
linked to lipolysis.
So that would be the other means by which lipolysis could be increased, is the increased sympathetic drive that occurs in heart failure. And again, getting back to this question, I don't know the answer to this, but is sympathetic drive higher in HFrEF? I'm guessing it is because it's a lower output state than it is in HFpEF, and that might also explain it.
I enjoyed your talks very much. So my question is that the acetyl-CoA product of the ketone, is it sufficient enough to actually change histone acetylation? Have you looked into that question?
So that was something, that was a whole half a year that we worked on, because people have shown both acetylation changes and beta hydroxybutyrylation changes in liver in response to ketones. And when we have done that either in isolated cardiomyocytes or in vivo when we do these infusions, and we at least look at RNA-seq readouts. We haven't done as much epigenomic stuff. We don't see any change, so we didn't go down that pathway. But others have shown exactly what you say. either from the beta hydroxybutyrate itself or the acetyl-CoA, but they've been in non-cardiomyocyte models, so for sure. I should also mention, well, I won't go into it. There's an HDAC part of that, but I won't confuse the issue.
Dr. Kelly, exciting talk. I'm wondering what decides the body to use the ketone pathway, for example, and exercise, you also need more energy, but under heart failure, you also need more energy. How the body decides to use the ketone and the pathological remodeling versus during exercise. So during exercise, do people also use the ketone pathway to provide extra energy?
Yes, they can. It's totally proportionate to, especially in the heart, to the levels of ketones that are being presented to the heart. So as a friend of mine, Maury Birnbaum, always says, that the heart is just stupid and the liver is smart, okay? And so it's the liver that really controls this. And whatever is controlling the liver ketogenesis is what's deciding it for probably heart and muscle. And in the case of exercise, there's a whole, there's the Cori cycle and many other types of things that can maintain bringing other substrates to the muscle. With that said, as you may know, the ketone esters, ketone esters first came out with studies that demonstrated, that were done at Oxford, that these drinks, these ketone drinks, markedly increased exercise performance. So if you're giving more ketones to the skeletal muscle and heart, the exercise performance goes up. They've also had to, they have to keep a close eye on this in bikers, et cetera, because you can buy it off the shelf.
Last question, Eric.
Really a little bit of follow Jerry's question, but I guess, we have natural experiments that are going on day in, day out with individuals who are putting themselves in ketosis for diet and other things. I'm just very curious if you've used those kind of natural experiments to understand the physiology.
I love these experiments. We haven't done them, and I'm almost always asked this. Another way to look at this is, what if we looked at intermittent fasting or timed feeding, where we know we have controlled levels of ketone bodies? I think that would be a phenomenal study in heart failure. That would be interesting to do. And, as I mentioned in the mice, we only need exposure for half the day. So it's not like you need 24-hour around the clock. So it's a great idea. Thank you.